Hi, my name is Zhu Liu. I'm a graduate student at the University of Southern California. Today I'm going to talk about AnnoQ, an integrated and interactive platform for large-scale genetic variation annotation. In the post-genome sequencing era, scientists who study disease are more interested in finding out whether there is any association between underlying genetic traits and the disease. This diagram shows association study in cancer research, which tries to see whether there is any variance with high frequency in cancer patients than the control group. If you are lucky, you may see this beautiful Manhattan plot. But the question is this the end of the game? The answer is not. Getting a list of variants is only the beginning of the question. What we want to know is whether these variants can be related to some pathways and process in a cell and tissue. Ultimately, we want to understand the functions they are associated with. In order to answer these questions, we have to first link the function to the variants, which is a process called annotation. For decades, the function has been studied in genes through wet lab experiments, such as biochemistry and molecular biology. So if somehow we can map those variants to some genes, we are able to link them with some function. For example, we may utilize the genome coordinate. If the variant is in some gene coding region, we may link to the genes and then link to the functions. Also, we may utilize regulatory elements. We can check whether those variants are in some regulatory elements, for example, the promoter or enhancer, and then link to the functions. There are also other studies directly link variants with disease and then link to the function. In the past decades, many tools have been developed for annotation. Most of them are focused on data in blue in the middle here. Also, most of these tools need bioinformatics skill in order to set them up and use them. You may need to use separate tools to obtain all of these function annotations. Each tool may have different system requirements. Also, they may design for different genome build. Installing all of these tools is a tedious work, and to keep them up to date could also be a nightmare. Here comes to our motivation. We are trying to build a comprehensive variant annotation system with the most up-to-date functional data and accessible to researchers with different research background. This is the AnnoQ homepage. You may access AnnoQ through the URL here, which is www.annoq.org. We organized all the resources related to AnnoQ in this homepage. AnnoQ is consisted with three main parts. The first one is the interactive query interface. It is a web page for users without bioinformatics skill. Users may directly use the web page to retrieve the variants with all of the annotations. The underlying background is an API. This API do not only support our graphic interface, but we also provide programming language packages which can communicate with this API. Here are some highlights for AnnoQ. AnnoQ is easy to use. No setup required, no computational skill are required if you use a web interface. Also, for users who want to do more complex work, we provide programming packages in R. The API has multiple functions. We also directly link comprehensive functional annotations with variants, such as gene ontology and pathways. We also utilize the enhancer to link variants with functions. With Paragene. Paragene is an enhancer database developed from another member in our team. This is how AnnoQ works. We gather variants into a dataset and pre-annotate those variants with existing tools such as WGSA and our own scripts. And then we import the data into a search engine we utilize Elasticsearch here. There is an additional layer for Elasticsearch for providing some additional functions for our API. People may retrieve data through our web interface or their own scripts. There are three major ways for AnnoQ. For users who only want to see a small number of variants and annotations, they may use a web interface. For users who want to retrieve large amount of data, the command line tools can be a better choice, as well as the R package. Also, due to there are hundreds of annotations in our dataset, users may select the annotations from the graphic interface and store this selection as a configuration file. The configuration file can be worked with our R programming package as well as the user's own created script. This is a list of AnnoQ annotation resources. Most of them can also achieved by other tools, except the blue ones. I'm not going to talk about the detail for those resources, but if you are interested in each of them, you may retrieve our documents through the link at the bottom. Thank you for listening. For other details about AnnoQ, you may refer to our poster in ISMB variant section.